Hi everyone, Dr. Hall here. The second part of the reproductive system. We're going to talk about how we make gametes and how those come together to form a new human. So part A of this lecture is going to be some introductory genetics, speaking in very broad terms, just so that we all have a basic conceptual understanding. Here we go. So it's all about the DNA, right? And you've probably heard people say, well, it's all about your DNA or your genes or your chromosomes. And you may have wondered, as I did when I was your age and even older, what is the difference between DNA and genes and chromosomes? Are those all exactly the same thing or are they different things? So we're going to kind of go through that. So the image on the screen right now is of the DNA molecule. It's called a double helix shape, which means a double spiral. A helix is a spiral. And so as you look at it, you can see it's almost as if someone took a really long ladder and then twisted it into this spiral. And you can see those rungs of the ladder moving upward. Let's take a little closer look at DNA. So DNA stands for deoxyribonucleic acid. It has to do with the structure of the molecule itself. So what I want to point out to you here, so if we look at the smallest level, we see that this molecule is composed of some sugars with some phosphate groups, and then something called nitrogen-containing base or a nitrogenous base. There are 4 different types: adenine, thymine, guanine, and cytosine that we find in DNA. Why am I telling you this? Why does this matter? Well, because these 4 different nucleotides, depending on what type of base you have, depending on adenine, thymine, guanine, or cytosine, is really important because these letters and the sequence of these letters ends up forming a code. So you can see how this is kind of the smallest level, and then here's an image of what that ladder looks like, and then that twists into that double helix form. So the arrangement of those nitrogenous bases, the adenine, thymine, guanine, and cytosine, A, T, G, and C, those are the 4 letters that we have to deal with, and they're going to be combined in different ways to form code and convey information. So a DNA molecule is really like a really long ticker tape. I don't know if you know what a ticker tape is or if you ever watched Addams Family. And so you can then read the code of the different sequences and arrangements of those 4 letters to get information. So that's why DNA contains our genetic information. So when we look at a double helix, when we look at a molecule of DNA, what we'll find is that certain regions of it have code for certain things. So in this example, I've said, well, maybe gene 1 has information that determines your eye color, and maybe gene 2, which is this segment of the DNA down here, is what's going to give you curly hair instead of straight hair, right? And maybe gene 3 further down on this DNA molecule has information on how to make dystrophin, a protein found in muscle cells. And in fact, it's genetic defects in the gene for dystrophin that cause the disease known as muscular dystrophy. See, it kind of makes sense. So genes are sections of the DNA that you can read the code, and it gives your cell information for how to do certain things that it needs to do in order to be a human cell. So for example, if we were using all 26 letters of the alphabet and a few symbols too thrown in there, right? Not all of the letters in this entire string or molecule of DNA, not all of the letters code for useful information or provide instructions to the cell, right? But some of them do. So in this case, some of these letters formed words, right? Hello, how are you doing? Each one of those words would kind of be a metaphor for a gene, right? It tells you how to do different things. So what's a chromosome? So a gene is a section of DNA that codes for a certain piece of information, certain bit of instructions on how to build or operate part of a cell, depending on what type of cell it is. The gene is a section of the DNA, and then our DNA is all coiled up and packaged into these little bundles called chromosomes. So a chromosome is a package of DNA, and a gene is a segment of DNA molecule that is code for a specific function or set of instructions. Here's another picture. I like this one too, right? So again, we have this long DNA molecule. It's ridiculously long. Different sections of it are different genes, different codes, different sets of instructions, and the whole thing is bundled together into a chromosome. So here are those definitions, right? So DNA is this molecule arranged in a long double helix, and it contains genetic information. A gene is the regions, the different regions of that DNA molecule that code for certain traits or functions, and a chromosome is just a package of that DNA. So when we look at our cells, all human cells, with very few exceptions, you might remember red blood cells have no nucleus and therefore no chromatin, no chromosomes, no genetic material at all. And then we'll learn about, later in this lecture, some cells that don't have the same number of chromosomes, but all human cells have 46 chromosomes arranged into 23 pairs. So there are 23 different types of chromosomes, right? So you've got chromosome 1, chromosome 2, we didn't name them very creatively. The only difference is that 23rd pair is called the sex chromosomes instead of being numbered. And different chromosomes have different strings of molecule on them with different types of genes, right? So certain genes are found on certain chromosome numbers. We have 2 copies of each of these different types of chromosomes. We have 2 copies of chromosome 1, 2 of chromosome 2, 2 of chromosome 3, and so on and so on. And again, all of our cells have the same chromosomes in them always. The difference in terms of how different cells behave and their structures are different have to do with which genes are turned on in those cells and which genes are turned off. Which set of instructions do they follow and produce and do, and which set of instructions do they ignore? So we have these 2 copies, right? And the reason why we have these 2 copies is one of them came from a sperm, from your dad or your donor or what have you, and then the other copy came from your mom or your birth mother or your surrogate, right? So sperm plus oocyte came together, and so you got one of each of your chromosomes for each pair from a sperm and one from an egg. Or an oocyte, same thing. So, and those are slightly different from each other, right? Because your biologic parents were different from each other and they had different genes, right? So in order to make a human, right, the sperm has to have only 23 chromosomes and the oocyte has to have only 23 chromosomes so that when they come together, the new cell, which is going to be called a zygote, has 46 in total, 23 pairs. So this is a picture of that, right? So gametes, and in this example, there's only 3 possible chromosomes to make it simpler for the diagram, right? So we have a haploid sperm. So haploid means half of the usual, right? So just 23 chromosomes usually. I remember it as halvesy, haploid. That works for me. And the oocyte also is haploid. And then they come together and they form a diploid zygote. So the zygote will then have 46 chromosomes, 23 pairs, right? So 2 of each. So what we're going to talk about next in the next section of this lecture is that actual process of producing these haploid cells, these gametes, these sex cells that have half the typical number of chromosomes.